Last time we talked about gene regulation in prokaryotes or bacteria. Today we're going to talk about gene regulation in eukaryotic cells. So this would be how genes get turned off and on in your cells. Now, if every cell in your body contains all 46 of your chromosomes, so contains the entire collection of genes, then why are cells different from each other? Well, not every cell expresses every gene, and not all of those genes are expressed at the same time. So in a typical human cell, only about 20% of the genes are expressed at any given time. And we call this differential gene expression, the fact that different genes are expressed at different times, and also different genes are expressed in different cell types. And we'll go into the details of that today. Now, in eukaryotic gene expression, we have a lot of opportunities to regulate how much of that protein is going to be active and expressed at any different times. And so these different opportunities are all different steps along the protein synthesis pathway. And so this slide here is a really important overview to kind of give you an idea of when you have opportunities to upregulate gene expression. That means to express more of that gene or more of that protein product, or you can downregulate it as well. We'll talk about chromatin modification, so making genes more or less available depending on the winding of the DNA, how much transcription is happening and how different genes get turned on, differential RNA processing, so that's going to be alternative splicing. And then when that mRNA gets transported to the cytoplasm, part of that involves the addition of the 5' cap and 3' poly A tail. It's going to get translated. Well, we have opportunities there to modify gene expression as well. How long the RNA sticks around determines how much protein is made from it. And also, once the protein is made, you still have opportunities to control how much it's functioning or how much it's working. So some proteins need special modifications to make them active or inactivate them. You can also target those proteins for degradation if you don't want them around anymore. So we'll go into more detail on each of these steps. Now remember that during interphase, so that's G1, S, and G2, the DNA is found as chromatin. And so there are two types of chromatin. We've got euchromatin and heterochromatin. In euchromatin, the DNA is not as tightly wound up, and it's more available for gene expression. So how do we make DNA more or less available depending on the tightness of the winding? We can modify histones. So remember that histones are the proteins that the DNA is wrapped around in chromatin. You can add methyl groups to DNA, which causes it to more tightly wind up and makes it less available for gene expression or for transcription. You can also acetylate DNA, which causes the DNA to loosen up and makes the genes more available. So let's take a closer look at this. So histones are the proteins that make up nucleosomes. So there are 8 histones within one nucleosome, and the DNA winds twice around each nucleosome. Now these tails here can be modified. So this is where we could have acetyl groups added on, for example. So these tails here can be modified. If you see these unacetylated histones, they clump together. There's a reason for this. There's typically lysines on these histone tails that have positive charges that interact with the other nucleosomes and allow them to tighten up. When you acetylate the histones, you add these little acetyl groups that are shown in green here. You're neutralizing that charge, and so you're reducing those interactions between the nucleosomes. And so they're now, instead of being attracted to each other, they're going to loosen up and have space in between them. So that makes the genes in this area more available for transcription. Now, you can also methylate DNA. Remember that methyl groups are nonpolar. So if you have a bunch of methyl groups on, you can actually methylate the histone tails, or you can methylate the DNA directly. So you can methylate like the cytosines in the DNA. And that's a more like long-term modification than just methylating histone tails, but they have the same effect. They're going to clump up because those non-polar methyl groups do not like being exposed to the aqueous environment of the cell. They'll clump up and we'll get heterochromatin again. So this is actually the basis of a upcoming or a newer area of research into genetic regulation called epigenetics. So epigenetics have to do with modification to DNA, such as acetylation or methylation. Now, there's a lot more to this, and a lot of it we don't quite understand yet. But what we do see is that chromatin modifications can be passed on from generation to generation. And these can be reversed, but we don't understand all the ins and outs of this yet. But this is one of the reasons why identical twins, although they have the same chromosomes, they have the same exact collection of genes or genomes, they're still different from each other. And that can be based on their individual experiences and environments, which can cause different epigenetic changes to their DNA. So different chemical modifications to the histones or to the DNA itself. So if you were to look at a picture of a chromosome, we usually see these dark and light banding patterns in the chromosome, and that's because they take up stains differently. Heterochromatin is the one that is more tightly wound. There's less gene expression in these regions. They tend to have a higher AT content, so they have more AT pairs than you would see in euchromatin. Euchromatin would have more GC content, and they stain darker. Okay, so this is the area that's more tightly wound up and less expression is occurring. So euchromatin, EU chromatin, is the area where we're going to see more gene expression from. So let's take a look at our next opportunity for regulating gene expression. And that's going to be initiation of transcription. And so how do we initiate certain genes, the transcription of certain genes in some cell types and not others, and also in different amounts? The more mRNA that you make, the more protein you'll get. And so we're going to take a look at control elements, which are regions of the DNA that interact with transcription factors and how they enhance gene expression. So let's take a look at how transcription initiates in eukaryotic cells. So we have control elements such as enhancers, okay? We also have promoters. Promoters a lot of times are also referred to as proximal control elements, or parts of the promoter might be referred to that. Proximal, think of that as being like in close proximity to the gene. It's nearby the gene. It's directly upstream. Remember, upstream means closer to the 5' end. Whereas if you were to look at the enhancer, okay, so this is another region in the DNA. It can be very, very far away from the gene. So we can also call that a distal control element. Think of it as being like it's at a distance from the gene. Now what happens is you have these activator proteins that bind to the enhancer. Okay, different cell types will have different activators present. And what's interesting is that we found that there are these mediator proteins. Okay, so this is going to be involved in recruiting RNA polymerase, so like transcription factors, that will bind both to the enhancer and to the promoter, although they're really far away in the DNA. And that is because the DNA will bend like a hairpin. So the activators present bind to the enhancer. Activators are proteins, enhancer is a DNA sequence. Once they do that, they help recruit these mediator proteins. You also have these general transcription factors that bind to areas of the promoter, and they interact with the mediator proteins. Okay, now all of these things coming together help to form the transcription initiation complex. They're going to be able to recruit RNA polymerase for transcription. Okay, so the fact that this bends in half in this hairpin shape, assisted by this DNA bending protein down here, allows it to recruit RNA polymerase and allows it to start transcription. Now, the reason that you get different genes turned on in different cell types is that enhancers will only turn on if the right if the right activator proteins are available to bind to the control elements of the enhancer. So in this example here, in a liver cell, you would have the activator proteins available that are specific to the enhancer found in this liver cell, and that would be able to form the transcription initiation complex. That's just the complex of the DNA and all the associated proteins binding that allow transcription to start. Whereas this crystalline gene, although it still has this gray activator protein that's required, you're missing the orange and the pink ones down here. So you need the right collection. It's not just enough to have just one of them. You need the right collection of the activators that bind those control elements, which are part of the enhancer sequence, in order to get transcription of the gene. So that's why you would have this albumin gene expressed in a liver cell, whereas crystalline, this is a gene specific to your eye, would not be expressed in the liver. Likewise, if you look at the lens cell in your eye, you'd have these pink, orange, and gray activators available. Those would bind the control elements of the enhancer in the crystalline gene, and that would allow you to form the initiation complex for the crystalline gene to be transcribed. Whereas albumin wouldn't be turned on because you're missing the red and yellow activators that bind these control elements in the enhancer of that gene. Remember that this dashed line here means that there can be a long stretch of DNA in between these areas. So the enhancer does not need to be close by to the promoter gene. You can also have repressor proteins that can bind and prevent transcription of those genes. There are also proteins that can recruit enzymes that help with acetylating or deacetylating, that means to remove acetyl groups from chromatin. And so that can also contribute to whether or not a transcription initiation complex forms. So I think that's enough for right now. So go back through these, take notes, try to memorize it, and we'll do part 2 of this slide set next time. Thanks, guys.